So I'm Dr. Esten Mason. I'm a professor and wheat breeder at Colorado State University. So if we think about crop domestication and the origin of plant breeding, so 12,000 years ago, there was this Neolithic agricultural revolution that took place in this area known as the Fertile Crescent. And so the first plant breeders were, and Rowan talked about this, but they were farmers, and they started to identify new traits, but it was really more of like a mutualistic relationship between the farmers and the plants in their field. And as humans started to farm, the plants had to adapt to have fitness within that those new farming techniques. So things like transitioning to a sickle harvesting system. You needed a tougher rachis to work to prevent shattering of seed onto the ground. So it needed to be harvested by humans so then it could reproduce the next year. Human dispersal of seed as opposed to digestion by a ruminant animal or something like that led to a thinner seed coat. And then our seed saving and then sowing that back the next year led to a loss of dormancy. So that seed had to be responsive the next year when it was planted in the ground. And so from this, you had centers of origin for different crops, and they would kind of spread out from those centers of origin. And so it's still debatable how many centers of origin there are. In this particular study, they're showing 15. But of course, you had wheat and barley, which were domesticated and had a center of origin in this Fertile Crescent region. Rice in parts of China. You had grains, sorghum, and millets in Africa, and then maize really in this Balsas River area of Central America, Mexico. And then maybe a second center of origin for maize in South America, as well as amaranth. And so crops were domesticated in these centers of origin and then moved out, across the globe after that. So I think to understand ancient grains, it's important to understand their evolution and understand the Triticeae species and how they evolved. And so there's a common ancestor that was present 10 to 14 million years ago. And there's kind of three branches that came off of that common ancestor. So you had rye, which in ancient times was sakali, but it's now just cereal rye, cultivated rye. And then you had another branch that was barley. So these are still obviously cultivated today. We all love and know barley for its malting qualities. And then you had this third branch, which eventually evolved to produce what we know as wheat, bread wheat, which is called a hexaploid. And so it's actually the combination of three different ancestral species. So it has three different genomes. And knowing this can kind of help us understand where these ancient grains come from. So what we would call the A genome is ancestral to Triticum monococcum, or one of the first cultivated grains, which was einkorn wheat. The B genome was closely related to Agelops speltoides, but then this einkorn combined with the speltoides relative to produce what's known as Triticum turgidum. And this would be a second ancient grain, wild emmer. So it has two genomes. In modern times, this is known as durum wheat. And so this is commonly used for pasta. So it has very high protein compared to other wheats. Then you had a combination of this Triticum turgidum or durum combined with what's called a goat grass, Agelops toshii. And that formed what we now know and produce as bread wheat. So it's a combination of these A, B, and D genomes. If we think about classification of ancient grains, I think, a way that we can really classify these is more just classified based on use or based on type. So if we think about classifying these based on type, we have what are known as cereals. So these are going to be monocot grass species. They produce a single-seeded caryopsis, or a seed that's very starchy. And so you would have things like einkorn and emmer and spelt as ancient grains, and then also rye, millet, oats, sorghum, barley, teff, and others. And then we have this other, oh, sorry. So yeah, just here's some pictures. So einkorn, emmer, spelt, and CIMMYT as well. And then you have this other group that we refer to as pseudo cereals that have really grown in popularity. They have really high quality protein. And these would be dicots. They're non-grass species, but their grains, their seeds do have some of the same starchy type of characteristics that we need, that we see in cereals and we need for baking and processing. So that'd be things like amaranth, buckwheat, and quinoa. We could also think of these based on gluten. So there's an interest in low gluten or gluten-free products. So gluten-free would be things like quinoa, amaranth, buckwheat, and chia. Gluten-containing einkorn, emmer, and spelt. So I see ancient grains as, being a plant breeder, there's a few different ways we can utilize these. So I see them as potential for using them as a genetic resource, but also potential for production and cultivation of these ancient grains. So one really good example of using ancient grains to increase genetic diversity is in this D genome. And so from genetic studies over years and years and sequencing of the hexaploid wheat genome, we know that this D genome is just low in genetic diversity. And so plant breeders have seen this as a way to bring in new diversity by collecting these landrace varieties, bringing them in and incorporating those useful genes, useful alleles into modern wheat. So goat grass, as I mentioned, is this D genome progenitor. It's present across a broad swath of the Middle East and Asia, Syria, and China. And it's the least genetically diverse. And so what we've been able to do is take a durum wheat and combine that with new sources of this D genome and make what we call synthetic hexaploid wheat. So this is non-GMO. This is just using breeding techniques, but we can bring in this new genetic diversity, and it's been used for things like increasing grain iron, grain zinc through what's called the Harvest Plus program, drought and disease tolerance, and other aspects of wheat. And these are just a couple of examples of just many, many success stories from bringing in this new genetic diversity from these ancient species. If we think about ancient grains for cultivation, so einkorn, emmer, and spelt, they're relatively unknown in the US on a large scale, but spelt would probably be the most commonly grown. Europe has cultivated these for some time in different locations. Switzerland, France, Italy, and the UK produce emmer and einkorn. In Eastern Europe and the Mediterranean area has produced spelt for a long time. But in the US, there's a growing interest in using these ancient grains because of their potential lower allergic response to glutenin and gliadin proteins. And so just as wheat has evolved, so here we have our wheat genome, we have an A, a B, and a D. As wheat has evolved, there's evolved multiple copies of these genes that control protein function when we make products from wheat. So these would be the high molecular weight glutenins, and this gives us our elasticity in that dough. And so there's kind of three sets of these. There's the glutenins, the gliadins, and then another group of gliadins as well on the group 6 chromosomes. And so there's been an interest in, can we use these ancestral wheat species, these ancient species, for potentially lower gluten content. And so I borrowed this slide from my colleague at Cornell, Dr. Mark Sorrells, who's a wheat breeder there, and he's had an interest in ancient grains. And so this shows just our ancestral species: einkorn, emmer, durum, spelt, as well as modern wheats. So those developed after 1950, and heritage wheats, those developed before 1950. So in heritage or modern wheats, you don't really see a reduction in these allergic properties. So we're looking at the relative celiac reactivity, and then we're looking at the presence of these alpha gliadins, which are the really reactive gliadins in wheat. So in either heritage or modern, you don't see this. But when we look at einkorn, we look at Immer, you actually do see a reduction in this response. You see a reduction in the presence of these alpha gliadin proteins. So because of this, Cornell, they've initiated a breeding program to try to improve the properties of einkorn and Immer, while maintaining that genetic diversity, but in improving some of the processing properties of these. And so, I think there's opportunity there to build on that genetic diversity. So that's all I have. Just like to show my contact information. We have our lab page here, wheat.colostate.edu. And this is my Twitter handle if you'd like to follow me on Twitter. And I will stop my share if you have any questions. Thank you.
Thank you, Dr. Mason. And yeah, I think we have time for at least one or so. If anybody wants to ask a question in the Q&A, feel free. I guess I'll just start though. I'm a little curious. You were mentioning, potentially using ancient grains for diversity and broadening that D genome. Do you have any other examples of, possibilities or things that are on the horizon with what land race varieties are used and kind of what some of that result is?
Yeah, I think, there's always been a need to conserve. I've served on as chair of the Wheat Germplasm Committee. And so thinking about where we need to beef up the collection, gene bank collections in the US and those type of things. Traditionally, it's focused a lot on disease resistance, bringing in new genes. And so a lot of times these wild or ancestral species, and even like perennial wheats and those kind of things, will harbor some unique genes that we can use for disease resistance. And now really climate change. So moving towards Aegilops, the D genome, Tashii, it's very tolerant to drought and heat stress. And so using that to combat climate change is kind of where we're moving.
Great, thank you. And I guess time for just one more quick one that I saw here. And it was just, I think, bringing up a little bit more of a broader educational question here about, can genetic diversity move us away from the original seeds?
Can genetic diversity remove? I think it's about figuring out what needs to be improved. So if it's obviously one of the things with ancient grains is the tight attached hull. So that's controlled by just a single gene. And so if you can identify genetic diversity in Immer for that trait, you could breed that trait out to make them easier to process. And so I think you can, genetics are such where you can target specific things to breed out or breed in specific things while maintaining that genetic diversity.